Bacterial connection.
Welcome back to Bacterial Connection. Last week, we helped E. coli find his lucky other half, and today we will help Helicobacter pylori find a soulmate. Hopefully. So, Helicobacter pylori, how are you doing today?
Just call me Pilly. I know how much of a mouthful that is. I'm doing great, looking for the one to spend the rest of my days with, someone I can relate with and make life miserable. Maybe, just maybe.
Well, hopefully, you will find the one today. Why don't you tell us a little bit about yourself? How were you discovered?
Well, Josh, I was identified by Australian scientists in 1982, Barry Marshall and Robin Warren. They found the bacterium, me of course, present in people with chronic gastritis and gastric ulcers, conditions which were thought as non-related. Of course, my boy Marshall knew the cure to get rid of me was through antibiotics. Don't tell anyone that, of course. But other scientists did not believe him since the stomach was believed to be acidic with no bacteria whatsoever, and dismiss his ideas since they believe ulcers form because of stress as well. Since I only affect primates, he could not justify his case using mice. So my homeboy Marshall got my great-great-great-great-great-grandmother, H. pylori, from an infected patient and made a broth which he then drank himself, proving his case. As days passed, he developed gastritis, the precursor to an ulcer, and he biopsied his own gut. He proved that bacteria were the underlying cause of ulcers.
Wow, that's some history right there.
You could say that. I'm also a proteobacteria, which means I am in the same phylum as E. coli and Salmonella, but I'm not as evil as them. I am actually closely related to Campylobacter jejungi, which is a common cause of food poisoning in the United States.
What other secrets do you have for us, Billy? Are you deadly? Are you gram positive or gram negative? Whatever that is, tell us more.
Gosh, Josh. Well, I am a microaerophilic bacterium, and that means I require oxygen to survive but low levels. I am also an invasive intracellular pathogen and have a curved rod helical shape. I mean, look at these wondrous curves. I usually penetrate the mucoid lining of the stomach.
Ouch! Who will be the lucky person today? I wonder.
Oh, it's not that bad. I have 4 to 6 flagella and am highly motile. I can also form biofilms. Since you're so interested, I am gram negative and I have an outer membrane unlike gram positive bacteria who lack it. Here, I'll show you on the screen. The outer membrane is a lipid bilayer composed of glycolipids, lipopolysaccharides, also known as LPS, to be exact. The phospholipid bilayer is inside. LPS is toxic to animals. This is usually an indicator of an infection.
Feisty, then I see. What symptoms do you usually give off?
Most people don't have symptoms, but when they do, it's excessive burping, feeling of being bloated, nausea, heartburn, fever, lack of appetite, or even anorexia, anemia, diarrhea, bad breath, vomiting that may include vomiting blood. I may be contagious, but most people already have my kind in their gut, without any problems. My exact route of transmission is unknown, but it most likely happens person to person by either oral to oral intake or fecal to oral intake, which that really means that it's likely waste-tainted water and is usually seen in families. I can usually be isolated from feces and saliva of infected people as well.
Well then, we set up Pilly with 2 people based on her preferences, and they were set up on a blind date during the week. So let's begin with the first blind date. Hi, Jared.
Hi, Josh.
We asked you both to score each other based on your first impressions. Here's how Pilly scored Jared. Now here's how Jared scored Pilly. So how was the date?
Well, I won't lie. It was a bit boring, but I got to know Pilly a little bit more, so it was not that bad.
It was not that bad. Oh, it was pretty bad, Josh.
I mean, you did talk to me about your antibiotic resistance and how you are hard to get rid of. 2 or more antibiotics are usually given together with a protein pump inhibitor. You also mentioned how clinthamycin is used to treat ulcers you cause and levofloxacin is used to treat infections. Talk about scary.
Hey, the reason for why I have become resistant to clarithromycin is because of 3-point mutations found in domain 5 of my 23S RNA gene, which is the most common binding site for antibiotics that inhibit translation. So it's not just me. Also, it creates a conformational change and leads to a decrease in binding of the drugs. The mutations occur by chance, so it's not my fault. And the same happens in levofloxacin, except that the mutations happen in different locations. These mutations prevent the inhibition of my chromosome replication, so it's not my fault, okay? Not me to blame.
Alright. Well, next up is Carl.
Hi, Josh.
We asked you both to score each other's looks based on your first impressions. Here's how Pilly scored Carl. Now here's how Carl scored Pilly. So how was the date?
I loved it. I explained how many people are asymptomatic, which means they are a carrier of a disease or infection, but experience no symptoms, meaning I may play a role in normal stomach ecology. I have also been found more common in developing countries.
That was definitely something interesting. I had a great time with Pilly. Diarrhea and vomiting aren't so bad. Plus, I might even be asymptomatic. How can Pilly survive in the stomach?
I mean, I have the ability to penetrate the mucus layer of the stomach, and I'm able to mount onto the surface of gastric mucosal epithelial cells. It then produces ammonia from urea. The ammonia secretion protects me by neutralizing gastric acid, allowing me to further replicate and further infect the cell membrane of the epithelial gastric lining of the stomach. Like this, I'll demonstrate. So as you can see, once I enter inside of the epithelial cell, if a lysosome attaches to me, then I am obliterated or gone, which of course I don't want that happening. But another way I can go is I invade the epithelial cell, and then I proliferate inside of the vesicle, so I multiply rapidly, and then I'm released, and boom. Because of this, gastric ulcers arise from the destruction of the mucosa, and besides the inflammation, mucosal cell death may also occur.
Further allowing Pilly to spread and deactivate epithelial cells that produce gastric acid.
Dang, he's so right. Then I'm able to escape immune surveillance and remain alive, repeatedly infecting other cells and recolonizing the gastric mucosa epithelial cells. And how do I survive, you may ask? Well, in order to avoid the acidic conditions of the lumen in the stomach, I use my beautiful flagellar hair to bury myself inside of the mucus lining of the stomach to reach the epithelial cells underneath where it is less acidic. I am able to sense the pH gradient and move towards the less acidic region. I adhere to the epithelial cells by producing adhesins which bind to lipids and carbohydrates in the epithelial cell membrane.
Just beautiful.
Well then, even though we're letting you know about your bio, I definitely feel the chemistry here.
If I may add, my outer membrane is composed of 5 major protein families. Adhesin protein, which are cell surface components that facilitate adhesion to other cells or to surfaces, and are a type of virulence factor. Porins, which act as a pore in which molecules are able to diffuse. These porins are big enough to allow passive diffusion, ion transporters, and flagellum-associated proteins, and other unknown proteins as well. Another important thing is my cytotoxin-associated gene A, which is also known as CAGA. Protein is delivered into gastric epithelial cells via a channel through which proteins or protein DNA complexes can be translocated. Virulence factors are translocated into the host cell. But one of the most important virulence factors invades the lining of the stomach and produces a cytotoxin termed vacuolating cytotoxin A, which is also known as VACA, and targets the mitochondrial inner membrane in the host.
So what is this VAC-A?
Well, Josh, it binds to host cells and is internalized, causing severe vacuolation, which is characterized by the accumulation of large vesicles that possess hallmarks of both late endosomes and early lysosomes. Apart from its vacuolating effect, it has recently become clear that VAC-A also directly affects mitochondrial function.
And her metabolism consists of fermentative and oxidative metabolism, lipopolysaccharide biosynthesis, nucleotide biosynthesis, aerobic and anaerobic respiration as well.
So in case everyone didn't know by now, pili causes gastritis and can possibly lead to an ulcer formation and even cancer, if not treated properly.
But I mean, who would want to get rid of me, right?
Well then, looks like we found your match, Pili. You and Carl will have a perfect getaway. See you next time on Bacterial Connection. Thank you. Bacterial Connection.